Hello everyone, I'm Xiaoting Fu from Ocean University of China. I'm working in the lab of seaweed chemistry and utilization in the College of Food Science and Engineering. Thanks for the invitation from Professor Allen, and thanks for all the organizers for giving me this opportunity to participate in this workshop.
My topic is about the biorefinery technology and products of carrageenan. It is done by me and Dr. Mu. The contents of my presentation including 4 parts. Part 1 is the carrageenan and Carotenophytes. Part 2 is about the industrial utilization of carrageenan. Part 3 is about enzymatic degradation of carrageenan, and Part 4 is about the enzymatic desulfation of carrageenan. So Part 1. Carrageenan is a natural carbohydrate obtained from edible red seaweeds. The name of carrageenan is derived from contrast crisps of seaweed known as carrageen moss or Irish moss in England. So the carbohydrates from carrageen moss is called carrageenan. And the carrageenan have 2 alternate subunits. Their structures are D-galactose and 3,6-anhydro D-galactose, with the possible ester sulfate groups in different positions of the saccharide units. And the seaweed produced carrageenan is called Carotenophytes. The red seaweed of Chondrus, Gigatina, Eucuma, and Carotenophytes are common Carogenophytes. Different Carogenophytes produce different kinds of carrageenan. This table listed the seaweed species and the relative carrageenan type. Their products are not pure. They are hybrid of different types, but with the majority of 1 or 2 certain types. For example, the Chondrus crisps, it produces kappa and alpha type, but for its tetrasporic, it mainly produces lambda type. And for Carotenophytes, it mainly produces kappa type carrageenan, and for Eucuma, it mainly produces alpha carrageenan. And here shows the structure of different carrageenan type. We can see that mainly the difference are in the sulfate group, the position or the number, 1 sulfate, 2 sulfates, and 3 sulfates. And the most valuable carragenes in terms of industry applications are kappa, alpha, and lambda. So we can see that different number and position of the sulfate group are very important for the carrageenan properties. If we want to better control its property, we should try to control the sulfate group. The part 2 is about the industrial utilization of carrageenan. Traditionally, the main application of carrageenan are in food industries. And because the kappa carrageenan can form the strongest gel, while the alpha can form a more elastic gel. And the alpha carrageenan has vast use in frozen dessert related to dairy products. It can give a smooth, creamier texture to the final products. And lambda carrageenan also is very soluble polysaccharide in both cold and hot solutions with very low concentration, and it can form a very high viscosity. And for the kappa carrageenan, it has a very strong water holding capacity, so it is very suitable to be used as a replacement for fat in the meat products. So it can improve the final texture. Carrageenan can also be used in the water gel applications such as the gummy candies, fruit gels, or fruit juices. The further increases in carrageenan use is in our life. For example, to use it as in the toothpaste or the firefighting room, the shampoo, cosmetic creams, some products like this. In the pharmaceutical industry, it also can be used to prepare the drug in the form suitable for application. And in this table, listed the changes of carrageenan market during these years. Various sulfated oligosaccharides possesses significant potential for the biomedical and physiological applications. For example, the sulfated galactone oligosaccharide from agrophytes such as Gracilaria, Phyrophora, and the fucoidan, and the sulfated alginate oligosaccharides from the brown seaweed such as Saccharina and Saccasome. So carrageenan, because it's full of the sulfate group, is a very important sulfated oligosaccharides. So it also have drawn considerable research interest during these years. So all these above sulfated oligosaccharides, they have the activities including anti-tumor, anticoagulation, anti-inflammation, anti-thrombosis, and the viral inactivation activities. Thus, the way to control the degradation and to get exactly the oligosaccharide we want, we must is very important for both its research work and the industrial applications, because we can see that different the polysaccharide backbone and different the sulfate group, the position and the number can affect its structure and its activity. So what we want to do is to get the compounds we want, but how to get it? We can use the biorefinery technologies. Both the biodegradation and to get the DP, which means degree of polymerization we want, and also to control the sulfate bond and to control which position and how much the sulfate bond exists, we can control by these two ways. So the first is the enzymatic degradation. The enzymatic degradation is done by an enzyme called the carragenase. According to the published papers, we can see that kappa carragenase and alpha carragenase are commonly studied and have published in various articles like this. In our research, we have isolated one marine bacteria which can degrade the carrageenan, and it is identified as a tubular species. The name is ZM2. After isolate this bacteria, we have done the gene cloning and the expression and also the product determination work. First is the gene sequence of this carragenase. And we analyze the gene information, including the homology, the conceived domain and active sites, and the signal peptides, and also to predict its structure and the catalytic sites. After get this gene, we further do the expression of its gene. First is in the E. coli. After the optimization of plasmid construction and the culture condition, the extracellular enzyme activity of the recombinant kappa carragenase was 25 times higher than that of the natural tubular, the natural bacteria. The products of the recombinant kappa carragenase was detected by LC-mass, and the main products were oligosaccharides of DP4, DP6, and DP8. For further application of the enzyme, Then the gene was expressed by Piscia pastoris. Piscia pastoris is a kind of yeast which is considered as a safer host than E. coli. So after optimization, the gene and the enzyme activity of recombinant kappa carragenase was 16 times higher than that of the natural strain. And the main products were DP4 and DP6. After we get the recombinant enzyme, we try to apply it. In order to comprehensively utilize the kappa ficus, we try to use the kappa ficus as the raw materials without isolation of its carrageen. So we use two kinds of enzymes. One is cellulase and one is our recombinant carragenase. Why we use cellulase? Because we have the cellulose in the structure, the cell wall of kappa ficus. So we first add the cellulase, and we can see the viscosity increase, which means the carrageenan released. And after 2 hours release of carrageenan, we add the recombinant carragenase in this point, and we can see the viscosity decrease and the reducing sugar increase. So after the reaction, we use different methods to prepare different fractions. They call the KS1, KS2, and KS3. We check the composition of the powder before the enzymatic reaction, which is called the KSP, and then compare with the KS1 and KS2 with it. We can see that the KS1 is the insoluble part with extra high concentration of protein. So we think it can be used as animal feed. And KS2, it is a soluble dietary fiber, so it maybe can be used in the food ingredient. KS3 is our main product. We want to produce the oligosaccharide, so we check its degree of polymerization were 6 and 8. So after that, we check the potential application of the products. First, we try to apply the formulated KS1 on shrimp cultivation. So finally, we find that the addition of 2% the KS1 into the shrimp feed can greatly help to increase the body weight and the weight gain rate, specific growth rate, and the survival rate. So it is a very good feed to the shrimp. And the effect of 3.5 kilodalton fraction on blood pressure were carried out in vivo. The results show that KS2 had a sustained antihypertensive effects on rat by reducing the level of renin and the angiotensin too. And then, in order to study the hypertensive mechanism, the effect on the intestinal health, referring to the intestinal flora regulation and the short-chain fatty acid were studied, and the results indicated that the 3.5 kilodalton fraction can regulate the intestinal microbiota of the hypertension. KS3 is the oligosaccharide of carrageenan. When carrageenan is used as polysaccharide, we pay attention to its viscous and gelling properties, while when it is degraded to oligosaccharide, there are diverse functions including antiviral, immunomodulation, anti-tumor, and cholesterol-reducing effects have been reported in various studies. So it is called functional oligosaccharide. Some of our former work indicated the anti-tumor activity and the immune regulation activity and the antiviral activity of the carrageenan oligosaccharides. Its inflammatory or pro-inflammatory activities have been studied because there are different molecular weight and different cell model used. So we can give also the results were varied, so it's very hard to give a conclusion. And also even for the same cell, but if the carrageenan oligosaccharides with different the molecular weight and different sulfate content, their effects are very significantly different. And some also give the conclusion that the low molecular weights with low sulfate content, the carrageenan oligosaccharides have the anti-inflammatory activity. So to better control the DP and the sulfate group, further researches are needed. To give a short conclusion is that by the biorefinery of by Carogenes, we can control the DP of oligosaccharide to make sure its activity and guide its application in different areas. So if we want to control the sulfate bond, we can use sulfatase. The carrageenan sulfatase have very high desulfation rate, which can remove the sulfate and form the anhydrous bond. Remove sulfate, form anhydrous. So the work is to develop different sulfatase with substrate specificities. Professors from Roscoff, France, discovered a novel alpha-carrageenan sulfatase from marine bacteria. It has been purified and used to control the desulfation of alpha-carrageenan. The result is very interesting that during the incubation, the signals attributed to alpha-carrageenan like this and this, and they are disappeared, and some new sites here, and here, the peaks appeared. And after a prolonged incubation, you can see that the initial alpha-carrageenan spectrum was replaced by a completely different spectrum of alpha-carrageenan. So the mechanism is predicted to be this remove. And in our lab, we have a very similar work. It's also a desulfation of sulfate galactone, but not carrageenan. It is agar, the desulfation of agar. And actually, the structure of agar is very similar to carrageenan. The only difference is that in agar, this unit is D-type, it's L-type, but in carrageenan, it is D-type. So our work is to find an enzyme which can remove the sulfate groups of agar and try to make agar to agarose. What we have done is publishing in 3 papers. First, we find the microorganism can desulfate the sulfate group in agar, and we isolate the bacteria and purify the enzyme. And secondly, we use the enzyme to desulfate and check the removal of sulfate group in both 1, 2, and 3, these 4 sites. And finally, we check the complete genome sequence of this bacteria to find there are 2 sulfatase and 2 aerosulfatase exist in this bacteria. So, furtherly, we will check whether this enzyme to clone these enzymes and to check whether it can also desulfate the groups in carrageenan type. And so, if we can biorefinery the carrageenan bisulfatase, we can first catalyze the formation of anhydrogalactose to improve the gelling properties of carrageenan. And then we can use this method to control and enzymatically synthesize new carrageenan structure. Those might harbor interesting functional properties. Let me give a conclusion. In the tropical area, we have so abundant resource of carrageenophytes. If we can use the biorefinery technology, we can get various kinds of carrageenan products that can be used in functional food, cosmetics, pharmaceuticals, and feed. So by this way, and the economic value of carrageenophytes can be greatly improved. And at last, I want to thank for the invitation and accommodation by Professor Allen and Professor Duan, and also want to thank the local areas to host this workshop. I like the work by Colony. And the two authors, I'm from the Lab of Civil Chemistry and Utilization, and Professor Mu is from the Lab of Applied Microbiology. We both from the University of Ocean, University of China, in the city of Qingdao, which is located in the north coastal area in China. Welcome.